Charles and Anthony, thank you so much for joining us today. And I will hand the screen sharing over to you.
Okay, thank you very much, Ken. So hi, my name is Charles, if you have not met me before, and this is my PhD trainee, Anthony Wu. So we're very privileged to be able to share our recent work with you, and I would like to thank Ken as well as the ISIF community for allowing us to share this with you. Essentially, something that we have been working on is multi-resolution imaging of extracellular vesicles as well as particles, and to elucidate what happens when we redirect the tropism within. So to go ahead, I know that this is a very specific crowd, but allow me to go over EV or particles. Is that, they come in many different types of subtype, including, if we go from the small one, exosome, that ranges from 40 to 100 nanometer. David Leiden's group used AF4, asymmetric flow field for fractionation, to further separate them into XOS, that's a term that they coined, or XOL. There are, of course, bigger ones that a lot of us are familiar are the microvesicles that shed off from the plasma membrane. So on the top are just images that we imaged back when I was a postdoc in Dr. Sandra Breakfield's lab. And here we actually label the cells completely green by using a reporter we developed called palmitoylated GP. And through live cell imaging, you can actually see that all these vesicles are being released. And if you isolate them and run them under TEM, and you can see the canonical lipid bilayer structure. So those are the ones that most of us are familiar with, that ranges from 30, 40 nanometers up to 1 micron. And of course, there are the apoptotic blebs that can be up to 4 micron, as well as oncosome from, from the Vesicles group that claim that vesicle as big as 10 micron can be released to help cancer cells migrate. Now, there are also researchers from China. They actually found there are pomegranate-like structure that release vesicles within, and they coined the term microsome, and they implicated that they play a role in development. And David Leiden's group in their paper also found there are particles, but not lipid vesicles, and they used the term exomer for sizes that are less than 50 nanometer. So these are all the particles and vesicles that we know today. And for the purpose of this talk, we're going to use vesicle casually unless we specify it. Now, of course, everybody knows that vesicles mediate intercellular communication, and they can be released through the fusion of the multipriscal body to release exosomes, or just shedding from the membrane to release microvesicles. I don't think we know how particles are released yet, but please correct us if I'm wrong. But all of these have been reported to carry bioactive cargo, including proteins, DNA, RNA, as well as lipids. And they can be actually delivered to recipient cells to offload this cargo to impart biological functions. And I think I'm going through this really quickly because this is very specific crowd. Now, vesicle definitely have been associated with almost any disease that you can think of, cancer, neurological, heart, immunity, infection. Well, everybody's under pandemic, so I'm sure there are many studies going on that. But one really hot field in vesicle is that people are trying to use vesicle as a biomarker, since especially for liquid biopsy, such that as if you look on the vesicle on the top right, that they carry all this bioactive cargo and they're being quote unquote nicely protected, supposedly. And so if people can harvest these vesicles and do characterization on these bioactive cargo, they could be very useful for diagnosis, especially for bedside diagnosis. Conversely, scientists are trying to use them as endogenous therapeutic vehicle to carry therapeutic cargo. And I think this is becoming very, very hot in the regenerative medicine field by using mesenchymal stem cell. So I'm just going again very quickly on briefly what we know about vesicle in terms of disease and therapy. There are many scientists who have done a lot of the early work, including using vesicles for immunotherapy. Or use RNAi. This is from Matthew Wood's group, that they use rabies glycoprotein for brain targeted delivery. And we're able to deliver silencing RNA to silence BACE gene. Now, there are other people who package vesicle or anti-cancer compound into the vesicles for anti-cancer therapy. So those are just an overview of what a lot of people are working on. However, if we're trying to use vesicle for therapy or to understand their biology, we really want to see where they're going. And at that time, when I started my postdoc in Sandra's lab, I was very curious. So I looked at the paper and then I found out, oh, okay, I think some of the golden standard technique include SEM, TEM. And as you can see on the right, these are SEM images by Dr. Johann Skok that I think this is one of the pioneer works where he scanned glioblastoma cell, which is a very aggressive type of brain cancer. And he found that there are little dots on the membrane. I think this slide will probably show many times. So I think Johan can start taking royalty on this. But in any case, this is what part of the initial work that led to the vesicle investigation from Sandra's lab, as well as me right now. So the thing is, we cannot image vesicle alive by using this technique. And of course, there is also atomic force microscopy and other ways. In terms of physical ways to study it, people have been using dynamic light scattering, NTA, zeta view, ISON. They each have its pros and cons. So due to time, I wouldn't go into the details. Now, molecular biologists such as us, we will use fusion protein or fusion reporter protein, such as GFP fused to CD63, which is a vesicle marker, or the more recent PH Lauren CD63 to detect the endocytosis. I believe this is from Lisa Weaver's lab, as well as some others. This is not comprehensive, so please forgive me if I cannot go through everything. And for other types of labeling, people have been using antibodies against vesicle markers, such as CD6381, or they can use dyes, such as PKH, dye-I, and the more recent MEMBRITE. Now, this is where I want to take us to, is that when I was in Sandra's lab, I was looking through the instruction manual, and this is something I wish to share with younger trainees, is that it's helpful to look through the instruction manual that is actually initially used to label cells for long-term in vivo imaging. And they are essentially aliphatic dyes that insert their very long hydrocarbon chain into a lipid bilayer. The longer they are, the more stable it is. However, their half-life can range from 5 days to 100 days. And at that time, I kind of raised a red flag because I personally don't think vesicle can stay that long inside the body. And not only that, later reports have shown that these dyes can actually form micelle, which is actually pretty bad because vesicle are already nanosized to begin with. And they also have dye residues. And as many of you know, every time you do an ultracentrifugation wash, you'll be really good if you only lose 30%. So washing them off is not easy. And similar principle apply to other lipophilic dyes. Now, we have read that MEMBRITE claim that they don't form micelle and then they are more specific. So maybe that's a better alternative, but at that time it wasn't available. And then, so we resorted to designing reporters. So on the right is just my extended metaphor, such that you're trying to dye your Easter egg, but you have to remember to wash off your dye. And that's actually a very labor intensive part. So we set a couple criteria at that time, is that could we label vesicles specifically? And in terms of accuracy, could we achieve spatial and temporal accuracy? Because from previous information on the dyes, we do know that the temporal resolution is probably really off. Especially, we are also quite concerned that the spatial resolution is not correct, because if the dye gets recycled by the cells, either in vivo or in vitro, you might not be tracing the vesicle anymore. You may actually be just tracing the dye. So those were things that we set for ourselves when we're developing the reporters. And in terms of resolution, I was young and reckless. So Sandra was she was just wonderful mentor to me. And she said, hey, just try it out. And I also have a co-mentor, Dr. Bakos Tanus. And then from that, we were actually able to achieve whole animal resolution imaging of vesicles as well as subcellular resolution. And just to go over really briefly, we developed the GLUC-B bioluminescent reporter to trace how vesicles are going around inside the animals after you injected them. And we were also very fortunate to collaborate with Mikhail Pitet's lab at Mass General Hospital with Ferdinando Pucci and Christopher Garris, and we're able to trace how melanoma release vesicles and how the body defend against it until they finally broke down and get controlled by cancer release vesicles. So we're very fortunate to collaborate with them. And we were also able to develop a fluorescent-based reporter that enable us to do subcellular imaging of vesicle release and uptake. So we're also very fortunate again to collaborate with Dr. Chen and Stott's group, also at NGH, and we were able to use those labeling technology to help develop a microfluidic chip for vesicle isolation from GBM patients. And from there, we can actually isolate cancer vesicles and then extract the RNA within for subsequent RNA analysis to do genetic subtype. And those were really wonderful work. And when I started my lab somewhere around 2017, we were thinking that, is it possible that we don't have to use two separate reporters to image vesicle? It would be nice if we can actually see all the way from nanoscopic resolution all the way to macroscopic resolution. And from here on, then I'll give the talk to Anthony, who will be presenting his latest work. So we're gonna do like a 21st century technology. We're gonna do a physical swap, and I'll give the microphone to Anthony.
So hello, can everyone hear me? And then yeah, okay. Thanks again for giving me this chance to present my our latest work in our lab. So as mentioned from Charles, we want to in this article or in this research, we want to develop a fusion reporter, all-in-one reporter that can use for the multi-resolution imaging. And this reporter has been used the BRET, bioluminescent resonance energy transfer, this technique as the principle to develop this reporter. And later we are going to use this reporter to develop study for the redirected tropism from a nano to a macro. So our strategy to develop this reporter is that we develop a palmitoylated PalmGRET. And this PalmGRET is the palmitoylated BRET pair, which use the facilitated EGFP and unlook as the BRET pair. And we want to use this reporter to anticipate this reporter can be used to study for the EP-based assay from a nanoscale all the way to a macro scale, cell-to-cell, cell-to-EP interaction, and then ex vivo biodistribution, and all the way to the macro level in vivo EP studies. And our design is based on bioluminescence resonance energy transfer. And we fuse the two proteins. Bioluminescent BRET is similar to the FRET, fluorescence resonance energy transfer, but just replace the donor fluorescent fluorophore with the bioluminescence reporter, which is the luciferase. And here we use the EGFP fusion with the unlook, which has been published in Shop and ET et al. Here. Yeah. And then after that, after the principle is that after catalyze of the furanzine, unlook catalyze the furanzine, this reporter can generate different modality for imaging. So the first is that after unlook catalyze the furanzine into furanamide, it will generate the first output is a bioluminescence. And after bioluminescence emission, it will the energy will resonance to excite the adjunct fused GFP, and then to have the BRET induced fluorescent signal. And this is the second modality. And the third modality we can use is directly use the external light source to excite the EGFP to have the fluorescent signal. And not only that, we facilitate a palmitoylation signal peptide and fuse it to the N-terminus of this BRET pair. And after post-translational modification, the palmitic acid will be modified onto the cysteine group of the palmitoylation signal peptide. After that, it will insert into the inner membrane of the cell. Upon expression, we are able to harvest the EVP expressing our PalmGRET. And not only that, we anticipate this PalmGRET to label multiple different, as previously introduced by Charles. We want to have its accuracy and specificity to the different pan-labeling ability to multiple EVP subtypes. So here we can see that after we transduce to have a stable expression PalmGRET into a 2,9-CT cells, we were able to, and this is based on the MySafe definition. We want to label the small vesicle, medium vesicle, large vesicle, and the recent subpopulation reported by David Leiden's group, which are the exo small and exo large and exomere, more separated from the small vesicles. And so first, we'll go through the characterization of our PalmGRET in the small vesicles. So after we isolate the vesicle expressing PalmGRET and the GRET. GRET is the cytosolic control to the PalmGRET. So after we use the standard protocol, and then after we apply, here we apply the 0.22 filter for the before the ultracentrifugation. And after that, we subject our isolated EV to the Schuker's gradient. And then first we harvest the different fractions. So after we, first we test the bioluminescent signal in a fraction by using the fluorescein. And then after here, we can see that the highest signal goes to SEV PalmGRET. And then the fraction we can see is from 3 to 6 fractions. And then later on, we further use the PBS to wash this fraction to further pellet those pellet from the fractions. So we can see that the signal is corresponded to the fractions signal, which can start from 3 to the 6. And after that, we further use the Western blot analysis for those pellets to see if our signal is actually from extracellular vesicle or a small vesicle. So here we apply Alex and then the CD63 as the EV marker to detect the extracellular vesicles, and then GFP to detect our reporter. So here we can see that in the left-hand side, here is the our SEV PalmGRET, and the EV marker and GFP are our reporter signal is coincide together, and then we can see that most of it is from 3 to the 6. And here also indicate that the GRET is our cytosolic control, which we know that the cytosolic part of the cargoes may randomly be packaged into the EVs. And here we can show that our PalmGRET perhaps packaging more or packaging our reporter or maybe the cargoes more efficiently into the EVs. So here we demonstrate that PalmGRET exhibit the robust bioluminescent activity in small vesicle, as we shows in the fraction pellet and then the Western blot. And after that, we want to see that since we claim that our reporter are labeled in the membrane of the extracellular vesicles, and moreover, we designed the inner membrane labeling since that we don't want to potentially affect the surface ligand receptor interaction between maybe during the attaching the vesicle to the recipient cells. So here we apply the dot blot analysis to detect that if our reporters to detect our reporters orientation. So here first is that we dot blot our EV gradiently onto the nitrocellulose membrane, and then by starting from the blocking, followed by antibody hatching and washing. We all separated into either two groups, either with detergent or without detergent. So here it shows that with the detergent group, the EV structure will be interrupted, and then the antibody can go in to detect what's encapsulated inside the EVs. And the data shows here. And on the other route is that if we did not apply any detergent, detergent here we use the 20. And then without detergent, the cargo or like our reporter encapsulated inside should be protected from antibody recognition. So here we can see that there should be no signal from the without detergent group. So as shows in here. So the upper panel will be the SEV PalmGRET, and then the lower panel will be a cytosolic control. So here we can see that with the detergent one, we can see we can detect a gradient signal from with detergent group, and without detergent group, it shows a very minor or very dim to a no signal from without detergent. And this suggests that our reporter are encapsulated inside, or it's labeled the inner membrane of the extracellular vesicles. And then the cytosolic control shows again that we can see that the PalmGRET palmitoylated reporter shows a higher packaging efficiency, or like it maybe has a more efficient way to be packaging into the EVs. So here we demonstrate that our orientation of the reporter is in the inner membrane of the EVs. And after that, we demonstrate our reporter specificity and then the orientation of our reporter. We then want to go through that if our reporter can label to the different populations. And here we first go to, since previously we used SEV, and then here we want to see that from report by David Leiden, there's exo small, exo large, and even the exomere. We want to see that we can further label the different population from these particles and extracellular particles, or like EVs. So we refer to the David Leiden's protocol. We use after we isolate the SEV PalmGRET or like GRET, we subject our EV to the asymmetric flow field fractionation AF4 to further fractionation different populations. And basically the smaller will go faster or be eluted faster. And then higher the size, it will be go low eluted slower. So here we can show, after that we go through the fractionation and put a different concentration, also refer to the David Leiden's protocol, and then test. After that we test the biodistribution signal and then the BRET-induced fluorescent signal activity by using the furanacid, the substrate. And after that, here, the upper panel shows the AF4 chart. So the left-hand side is the GRET, and then the right-hand side will be the PalmGRET. And we can noteworthy part is that the P2, P3, P4 one is stand for the exomere, exo small, and exo large population. And noteworthy part is that we can see that in the PalmGRET, it somehow increased the size in the exomere part compared to the cytosolic control. And then after that, we harvest, we pulled all the fraction and concentrated it, and then tested with the bioluminescence signal and then the BRET-induced fluorescent signal. So here we can see that there's a robust signal from the exomere, either from both exomere, exo small, and exo large. So here we demonstrated that our PalmGRET also labels the different subpopulation in the small vesicles. And not only that, after go through a small one, we want to go to a bigger one to see if our labeling it's like a different ability to label the different populations. So after that, we refer to the general protocol for isolate the median large vesicle, which may use the 10K pellet technique for isolation the median large vesicles. And after that, we use the same supernatant to further subject to the 100K to isolate the small EVs. And then the other route is that we, for our later experiment, we use the 1.2 filter to filter the condition median. And after that, we subject it to the 100K and then to have a 1.2 micron extracellular particles. So here is the NTA size distribution data. So here we, from the left to the right is that the 10K pellet stand for the median or large EV, and 100K pellet small EV, and 1.2 micron filter extracellular particle. So here we can see that in the large particle one, we can see there's a higher portion of the percentage of EV particle in more than 200 nanometer. And the 100K and 1.2 micron filtered SEV or EP, they shows the similar size distribution or percentage of EV particles. And then it shows a higher portion to the smaller than 200 nanometer. So here we can see that our PalmGRET labels the multiple EVP population based on our isolation method. And then it shows that the median or large and then small vesicle and then extracellular particles. So after all these EV characterization, we know that our reporter has the ability to pan label the different EV subpopulations. And later on, we also test the bioluminescence and BRET fluorescent signal. And then we can also, this again shows the PalmGRET has a higher signal than the GRET. And then here we can see that in the RUU per EV particle, the micro, medium, or large vesicle can exhibit or like shows the higher signal compared to the small vesicles, because the small per particles. So after we demonstrate the characterization of all the EVP image characterization for our PalmGRET, we want to see that if our PalmGRET can actually be checked lively in the cells. So here we apply first we apply the confocal to detect the it's like a distribution. So the green one is our reporter PalmGRET, and then the red one is the published first generation reporter published by Charles is the TD tomato. And here we can clearly show in the 3D imaging. We can see that there's the two population. We can clearly imagine the two population of the EV lively. And not only that, we apply. Not only that, later we apply the super resolution imaging, which is the SURF. The SURF stands for the super resolution radar fluctuation, and this is a technique for the super resolution nanoscopy. So here we can see that if our reporter can actually stand for, our reporter can be tracked in nanoscale in nanoscopy level. So upper panel one, we can see that the lower panel one is the epifluorescence. The barely distinguished two dots or like signal can be clearly distinguished from the SURF. So here not only shows that the ability of the SURF, but also shows the ability of our PalmGRET can be used in the nanoscopy imaging. And after this imaging, we also will be able to check the lively. We are able to real time check the track the EV protrusion-like signal from the nanoscopy, the SURF imaging. So here is the signal we are able to track from the edge of the cells, 293 PalmGRET cells. So after we go through the nanoscopy imaging, and then we want to see that after in vitro study, we go through that if our reporter can be checked in the in vivo imaging. So here we are using the labeling 293T. After we harvest the PalmGRET EPs, we inject the EP into the C3H mice, and then we use a 100 microframe for proof of principle. And after that, after 5 minutes post injection, we use the fluorescein. We all use the tail vein injection, IV injection, and then we take the imaging from 5, 10, and 20, all the way to 30 minutes. So first show is the bioluminescence signal. We can detect the signal up to 10 minutes, and then barely detect it in 20 minutes. And then suggesting that our reporter can have a long-term tracking ability. Also since this will be more convenient for studying the immunology compared to like previously the GLUC also has a very good signal, but the GLUC's handling time is very short. It requires a very high technique to or like very specific time course to detect the signal. And after that, we apply the filter to detect the BRET signal induced by BRET. So here we only apply the filter. We did not apply any external light source, and we can see that there's a more clean signal here. It's from the BRET signal. And after that, we also use the excited for laser to excite our reporter, and we can see that the signal is totally off compared to the bioluminescence and the BRET signal here. And after that, we use the we harvest the organ from those 100 microgram injected mice. Here we can see that the BOI BRET, again, the signal is highest in the bioluminescence signal and then the BRET. And the excited fluorescence also shows the off signal compared to the bioluminescence signal and then the BRET induced fluorescence signal. And here we can show that most of the injected EVs goes to a lung first and then followed by spleen and then liver. And not only that, we also, after the macro in vivo and then the ex vivo biodistribution, we also do the cryo-sectioning on those injected mice. And then we do the cryo-sectioning and immunohistochemistry after, and then followed by immunofluorescence staining. And then we are able to detect the extracellular our EPs in the tissue, lung tissue here. But we also did the different other tissue for the quantification. And here we can see the merge channel with the merge channels with the DIC and then the DAPI. And after that, we also used the ImageJ to quantify 70 images from those cross-sectioning. And we were able to quantify our EVPs in the different major organs. And here we can see that in the right-hand side here is that the highest signal goes to the lung, followed by the liver, followed by the spleen, and then liver. And then the kidney was used as the negative control. And then here we can see that this imaging just shows a reminder that the highest goes to a lung, and then followed by spleen, and then liver. And the kidney shows the barely a signal or a background signal. Yeah. So after that, we want to after we demonstrate that our reporter's ability from the different modality and then for multi-resolution. So we want to apply our reporter to see if it can report actually the different biological event in vivo or like in a biological context. So here we all know that has been reported by that also by David Leiden's group that the tumors has its organotropism from a primary tumor either goes to the lung or even goes to the liver. But however, most of the time we only during the study we will focus on a specific organ. But how about the different organs such as the brain and then heart, spleen, liver, or other like major organ, kidney? So the biodistribution most of the time are missing during the specific study on those tropisms. So here we want to see that we want to see that our reporter can actually report the biodistribution in these by using biodistribution in for our extracellular or like in EVP studies. So here we establish our PalmGRET in different model. Here we use the HCA1. This HCA1 is the murine hepatocellular carcinoma derived from the C3H mice. So here we established our HCA1 PalmGRET, and then we also topically implant the HCA1 PalmGRET into a C3H mice, which is immunocompetent, and then in the liver. And then after in the week 0, and then we harvest imaging and harvest the organ for the in vivo for a study for IVS imaging at week 6. So here is the week 6 imaging in vivo and ex vivo. So here we can see that it reports the majority of the our liver cancer here, and then both shows in a bioluminescent signal and a brain-induced fluorescent signal. And then after we harvest the organ, we can see that this is a liver plus the tumor signal. And since this tumor HCA1 has been reported to has the spontaneous lung metastasis, so that's why it's a ideal model to study for the metastasis cancer biology. And then here our reporter can also shows the spontaneous metastasis signal to the lung. So after this one, we want to see that since the tumor the cell shows this chain, we wonder that if the HCA1 derived EV also shows the tropism that can goes to the lung. So here, after we harvest the EVP from HCA1, we inject the 30 microgram we educate the C3H mice with our EVP HCA1 PalmGRET. And then we inject 4 dosages, which start from 0 hour, 24 hour, 48 hour, and 72 hours. And then we harvest the organs at 0.5 hour and 72.5 hours. And after that, we were able to have the pie chart to detect the different percentage of our EVP distribution. So interestingly is that at the 0.5 hour, most of the our particles goes to the lung. And then even after 72.5 hours, it still has the highest portion goes to the lung. And here we suggest that this reporter or this HCA1 derived EVP has a long tropism. And after that, we want to see that what's the protein or like what's the content that direct the long tropism of the HCA1 derived EVs. So after we isolate our EVPs, we send it to the proteomic analysis. And we were able to, and after the gene ontology and then the different literature search, we were able to we were able to detect, or we were able to select the different candidate from. They are mostly the membrane proteins, which are the SLCO2A1, NHER, NERF1, and then CD13 or CLIC1. And the function for the criteria for searching this membrane protein, first is a membrane protein or like membrane associated protein. And then the other criteria is that it has been reported to related to the liver cancer progression or due to maybe the lung microenvironment change. So after that, we want to see that if we can do any like modification or like knockdown on these candidates to see if we can actually redirect this tropism of our EVPs from HCA1 PalmGRET. So after we knock down of these candidates from the HCA1 cell, we harvest the EV and then use the same time course to educate the mice with the different knockdown group. And then here we can see that this is the time scale we use. And then we further reduce the injected dosage to 10 microgram here. And then we, as the previous time course, we educate the mice with the 0 hour, 24 hour, 48 hour, and 72 hour, and harvest the organ at 0.5 hour and 72.5 hour. And these are the group we use and scramble as the negative control. And then here we can see that as the percentage of the detected signal in the lung, we can see that in 4 out of 3, which are the SLCO2A1, CD13, and CLIC1, shows the reduced or we redirected the tropism, the reduced lung tropism in the lung. Here we can see that compared to the scramble one, the SLCO2A1, CD13, and CLIC1 shows the reduced distribution to the lung. And then liver is the other major organ we are going to study. And then here we can see that the CLIC1 and then the CD13 shows the initial increasement in the liver. And not only that, we also investigate different organs distribution. Since we previously mentioned that not only we can see the major redirected tropism in a specific our target organ, we can see that the different organ has its own different distribution, redirected or like a biodistribution after the education and then the injection. So here we can see that in the spleen one, we can see that in the SLCO2A1 it shows the initially increasement, and then also in the CLIC1. And then after 72.5 hours, the CD13 knockdown shows the increasement as well. And kidney here is interesting is that SLCO2A1 shows the initially increasement and then followed by the 72.5 hour increasement as well. And then overall this data suggests that not only we are able to redirect the tropism of the EVP, but also we can demonstrate the different biodistribution, distinct biodistribution profile from the different organs, such as the liver, spleen, kidney. And the heart and brain shows the very less amount of stand for the very small amount of the percentage of the detected signals here. So overall here is that our PalmGRET reporter is enabled to enable the inner membrane EV membrane labeling. And then followed by, we here demonstrate that we cannot say that we label all the EV population, but here we have the extracellular particle labeling. Start from based on our isolation method is from exomer, exo small, exo large to the medium and large EVs. And then the third one will be our multi-resolution EP imaging. And then not only the multi-resolution, multimodal imaging, we were able to do the quantification in the tissue section here. And then lastly, our reporter are able to report the redirected tropism and then distinct biodistribution profile. And not only that, since our PalmGRET is, in my opinion, is quite powerful for the study for biodistribution. So here, maybe in the future or like in different cell, like a therapeutic strategy, the PalmGRET can help to study for the different distribution of the therapeutic effect of the EVP as a therapeutic target. So here I want to thanks again for the inviting talk to share our recent latest work in the EV Journal Club. And I want to have a big thanks to my mentor, Charles. Without him, he gave me a lot of support and then a very constructive instruction and then a lot of guidance. And then thanks to all the lab members and then collaborators, and without them, this wonderful publication cannot be published. Yeah, and then here I will want to switch the channel to Charles again.
So this is just like a 10-second advertisement that we are recruiting. If you're ever interested in us, we are located on this map right here, right smack in the middle. And if you're interested, scan the QR code or visit our website. And that's it. I think that's my 10-second. And finally, I to thank all the audiences for your attention, and we'll be happy if we can entertain any questions. Thank you.
Well, thank you very much, Anthony. Thank you very much, Charles, for that clear presentation. And thank you for all of your work that has gone into helping the community, trace these vesicles that we're so interested in. I think it's also great. I just want to mention that Dr. Zandra Brakefield is on the call here today too. So we have 3 generations of scientific training here. And it's great that you're recruiting for your lab too, Charles, to expand. So we do have quite a few questions that have come in already in the chat box. And just as a reminder, if you do have a question for Charles or Anthony, please use the chat box so that we can let you unmute and you can ask this question. So I'm just going to change the setting here so that people can unmute themselves, and then we can start with some of these questions. So some very good questions here. Let's, when I call your name, please go ahead and unmute yourself and you can ask your question. So we'll start with Shima Gorogi, who was actually a recent presenter here on the EV Club. So Shima, go ahead with your question.
Hello, everyone. Thank you for your nice presentation. I think I asked my question too early and I got the answers in the later slide, but thank you. It was such a great work.
Okay. So you had asked the question about the harvest. Yeah, so that was.
Yeah.
And next is Peter O'Brien. So Peter refers to a paper that was published some time ago with quantum dots and looking at quantum dots going to lymph nodes. So Peter, go ahead with your question. Did you look at the lymph nodes? So what are your thoughts on lymph nodes and what's happening in them?
I did not look into the lymph node, but since to us is to me or like to us is like it's possible to harvest the lymph node, but I think that it's possible to detect it's possible, or maybe it can go to the lymph node, but in our setting we did not go to the we didn't harvest the lymph node and then to see the actual signal in the EVPs if our signal is there.
So just to add to Anthony's point, so no, we did not look into lymph node, but we do believe that the reporter can detect it, because when we were using the earlier generation, the GLUC-B, we collaborated with Mattel lab, and they were able to harvest the lymph node to detect the amount of vesicle within. Yeah. So, and then with the current reporter, the signal is even more long lasting. So we would say that it definitely increases the chance of detecting it. Yeah, even in minute amount. The fact that we can detect a little bit in the brain was very encouraging. Yeah.
Great. Okay, so we have some questions that are closely related coming from Fabrice Lucien and then also from James Drummond. So Fabrice, why don't you start here?
Yep, thank you. Thank you, Charles and Anthony. That was great presentation. Very nice data. So the first question that I have was actually, I think you presented in one of the slides, is that you can image EV distribution by serial injection of the compound like furomycin, right? So you can do like multiple injection over the time to try to light up the EVs more you progress into the course of the experiment.
Oh, very good question. I'm gonna give this to Anthony. Yeah, he had tried this.
Actually, I've tried this like before. I even like I can use the multiple dosage of the furosemide to light it up again, right? Yeah, I did it before. Yeah, but it still requires since if after the distribution or first injection, the EV will go into the deep organs. So in the in vivo imaging, it may like through a time, if you use the second dose of furosemide, you will light up again, indeed, but maybe you will have a less signal, and then maybe third dosage, it will disappear since there's a time course for either the reporter or the furosemide imaging depth of the iris. Yeah. So it's possible because after we harvest the organ, we can do it like multiple trigger for the signal. Yeah. After we homogenize the organ or even do the ex vivo, we can apply the immerse into the diluted furosemide, and then it can be imaging multiple times. Yeah, it's possible.
and
so does that, sorry, let me just ask James. James, does that answer your question too, or do you have a follow-up?
Well, it does. Yeah, I mean, I can understand you want a stronger signal. I just was concerned that you're maybe saturating certain systems and driving it into other corners of the body because of that, or I just would like to see what would happen after one dose. Where does it all go?
You mean one dose of vesicle or one dose of the substrate?
One dose of vesicle.
Very good question. I'm gonna hijack this question if I may. So for the education experiment that we did, we actually lowered the doses down to 10 microgram. Yeah, so like I know that it wasn't very obvious. So we actually lowered from 100 microgram to 30 microgram to 10 microgram. So for the tropism study that you saw at the last part was actually done at 10 microgram. What we realized from our previous experience is that if you inject with a large dose of vesicle, they actually get pushed to the spleen. We don't see the spleen anymore at 10 microgram if you look at our paper. And that's actually very interesting because we didn't know how much is actually more quote unquote physiological relevant. And back then, the paper we published with Sandra on ECS Nano, that one we injected with 100 microgram. And then they went to the spleen. And then we washed the spleen. None of them actually got retained the spleen. So that was very interesting. So our hypothesis was that vesicle actually gets trafficked to the spleen as a temporary reservoir, because spleen is a highly vascularized organ. Mm-hmm. And here we show that by injecting at only 10 microgram, the spleen didn't gather much vesicle anymore. So it kind of answers your question indirectly, I hope.
Sure. Yeah. Very good, thank you.
Yeah, very nice question.
And back to you, Fabrice. Sorry to interrupt.
No worries. Actually, you were on a good way in your answer. So what's the lowest concentration of particle injected in mice that you can detect with either BLI or BRET, fluorescence? Can you go down to 10 micrograms?
Yeah, I tried the different doses such as the 100 microgram, 50 microgram, 30 microgram, down to the 10 microgram. The signal, the best signal we can see, not only based on expression, since tunnelity is quite a good expressor, and then either we use so proof of concept we use 100 microgram here. 50 microgram I can see that, but down to the 30, barely to no, and then 10, I cannot detect signal from in vivo. Yeah, okay. Directly use the live animal. Yeah. But 10 microgram can be detected after homogenize the organs. So that's a powerful for the biodistribution study. Mm-hmm.
Yeah. So Fabrice, just to add, like it also depends largely on the expression of the reporter, right? Mm-hmm. So you have to play, because any reporter, if you overexpress it, your cells are not happy. So you kind of have to find the best compromise. And here we did HCA1. The expression is superbly low with our reporter, and it still works. So I don't know if that adds some confidence to you, but that's something that if you like to try, just keep that in mind.
Yeah, if I can add a last element, is that I don't know how the sensitivity of GFP to the pH, acidic pH, can also affect the fluorescence in vivo. Since GFP can lose some emission, some strength based on the acidic pH. Just to note, that's maybe something that could also
Yeah, that's a very good point. In fact, sorry, I'm going to hijack this one. You know, so we actually did do baseline for all of the biodistribution, meaning that for the BLI or the BRET signal, those were all normalized against the background before we actually put it into stats analysis. So that's something that we also emphasize on, because bioluminescence reporter are prone to auto-oxidation, and different organ has different stage of oxidation. So it's important to do that. GFP, yeah, we didn't really pay too much attention to it because the primary source of signal was actually from the bioluminescence, right? So yeah, but I do see where your question is coming from. So it's important to include the negative control, the background controls for that. Yeah.
Thank you guys.
Thank you.
Great question. So I like this discussion, and I want to ask a question now from Tom Dredunk, who I think had to step off, but he asks if it is possible to do imaging in tissue section. So can you just section this tissue and then put the furamazine on it?
Since I think that in our setting is we use the 10 microgram for cryosectioning, and then for 10 microgram is that we have to consider the whole organ mass. So after sectioning, it's just 5 micron or something. So after adding the furamazine, it's very hard to detect. So that's why we use the immunofluorescence to add up the signal and then to detect it. Yeah. So if the 100, I don't know if the 100 microgram can help, but because the tissue sectioning shows in vivo all the way ex vivo and cryosectioning and to quantification, that one is used the 100 microgram. And that one we still need to add up the immunofluorescence to add out a signal. So if it goes down to the 10 microgram, I don't think it will work. So directly active furamazine one, I did not try, but I believe that it's very hard to detect since or you need very super good detector. Very good detector, which I believe that it's not possible to be detected now since it's very weak compared it just in the 5 micron meter of the stick of the slide. Yeah.
Let me add to this question.
Here. I'm sorry,
yeah,
Tom.
Hi, Tom.
I'm here. Sorry, I had some internet problems just now, but I'm back. So the reason I'm asking this question, nanoluciferase is super bright compared to any other type of reporter. So I thought it may be possible, but I had some troubles with it myself. So I was wondering, if you succeed.
We don't think as Anthony explained, because if you imagine an organ, right, and if you cross-section it, you're literally dividing it by, say, more than 1,000. So, and then to visualize it by detector, it's really difficult. Even if you have an EMCCD, I would say it's really tough. Yeah, so personally, I don't think it's readily feasible. Yeah, and
I'll go for labeling then.
Yeah, but maybe something that we can point out is that the reason why we did all these imaging is actually to do orthogonal assay to validate the reading. Right, so because if we were only to see the signal in vivo or ex vivo, then you might challenge, right? Then we do orthogonal assay by using ICC, IHC, and then we quantify it and show the same trend. That's why we did all that to help. And the next step that we're trying to see, and I'm sure these are questions people have at hand, is what cells actually took them up once they get to the organs, right? And then those are the next thing that people can use the technology for, or even fact sorted. Yeah.
Yeah, very good.
Thanks.
So I want to get into a question by Eric Boilard. Eric, do you want to go ahead with yours?
Thank you, Ken. Hi, Charles and Anthony. Thanks. It's a fantastic work. I was wondering if, when you inject, I think you injected in tail vein in the mice, and I would think that the lung vasculature would be the first vascular bed that is highly vascularized, and there are very small, tiny blood vessels, and whether you think that the first organs that the EV will go through will impact their tissue localization, and whether you had ever compared the impact of the route of intravenous injection, for example, vein or aortic or intracardiac injection.
We did not try the other routes for injection, but there's a paper that published in JAVE that using they have tried a different routes and then different dosage, and then use the dye to DIR. I believe that is DIR. I remember it's DIR dye, and then it shows that the majority goes to the liver first. And then in our study, our reporter shows the long tropism, and why we can say it's long tropism is that we also use the 293T, though that's not a very good control, but we use the 293T as a control to see that after education, it did not like increase its like distribution to a lung after 72.5 hours. So that's why we claim that our like distribution to our HCA1 PalmGRET EVP has the long tropism. So yeah, indeed it accumulates more in more vasculated structure, such as the spleen and then the lung. But after our because we have done our control, so so that's why we claim that our EVP has a long tropism. Yeah.
Yeah, sorry. So basically, long answer short is we didn't try it. Yeah, but I think it also depends on the route. It also depends on the dosage amount. Yeah. So sorry, we have not tried that. Yeah, because we have already used a lot of mice.
Yeah, I can understand that. Because we actually injected same amount of vesicles that were much larger than yours, but I can see there were vesicles, and we compared cardiac and tail vein, and tail vein was lungs and cardiac was kidneys. So that really impacted their distribution, whether they were injected in different.
Yeah, I definitely think the injection route would affect the biodistribution. I think that's taken for granted. I mean, even with nanoparticle work that people have done with liposome and stuff, they also show similar trend that the biodistribution is administration route dependent. Yeah, but note the dosage. I think that's something that a lot of us miss. Yeah.
Okay, thank you.
We have several questions too, and I think we're coming towards the end of the hour, so I'm just going to summarize a little bit here, and maybe we can just have a brief discussion on this. And I think it comes down to whether there are different biodistributions of different classes of EVs, whether exomeres are going to different places, large EVs are going to different places. So Esther Naltatun asks about this. Sand HUJ, I'm not sure how to pronounce that, is also asking about whether things are stuck in the vasculature or whether they're actually going across bodily barriers like the blood-brain barrier. And yeah, so do you have any evidence maybe that exomeres or large EVs or whatever are trafficked differently?
We are definitely working on that. It's ongoing for sure. We speculate that it will differ. We're not sure yet, but so we don't want to say too much. Yeah. So, but we do have the tool. So it's ongoing work. So sorry, I think that's as much as I can say based on our data. Vasculature wise, I think it goes back to the points that we had I think it also depends a lot on the dosage. Yeah, I think people need to be aware of the dosage because previously we really didn't know. So we were injecting 100 microgram, and then we kind of say, wait a second, this is way too much, and we started scaling down and down and down, and then yeah, that actually did affect the biodistribution by quite a bit. So I think that's something to keep in mind, and it may address questions such as does it cross the BBB, or are you just forcing it through to some extent? Yeah.
So that was my question. My name is Jagdeep Sandhu. I'm from Ottawa, the National Research Council in Canada. And
Hi.
We are like as fascinating as the EVs are, we do work with them, but I find it is very difficult to really see them. One of my graduate students is working on the, like really visualizing them, where they are going. We work in the area of blood-brain barrier, and it is like a nightmare to do any kind of imaging. So she keeps asking me, how much should I put in? Like we count and put some EVs, right? So I don't have any standardized method to do that. So how do we know what amount we are going to put? What is its fate in vivo? Where do they really go? How much goes to the lysosome? I have no idea. Like we are just testing some things, but I think the field, there is no standardization and guidelines. So it's really very difficult for us to work actually in this field.
Right. We share the pain with you because, actually, by the way, I grew up in Canada. So hello. it is true. We don't know what is quote unquote physiological relevant dosage. We at least I don't know yet. But at least we tone it down to 10 times less now. Can we tone it down even more? I think with our reporter, we can try. We just have not tested the limit yet. But I do know that the rabies glycoprotein, that the coding by Matthew Wood's group, I think that was published in 2012 or 2013 in Nature
2011.
Biopack. Oh, 2011. Sorry, I'm aging. So that I believe have been repeated by some of my colleagues to actually target a brain. Okay, they were using dye to see that. But again, that's as far as I know. That, and then I believe other people came out later to either use was it aptamer or peptide to also target a brain with success. Now, the dosage that they use, and I believe Ken can resonate on this, is that since there's no standardization on the amount you put in, some people use microliters, some people use counts of particles, some people, for our case, we use concentration in terms of protein. So it's really hard to, you know, compare them side by side. And I think that's, hey, maybe Ken does MySEF 2021 or something that we can put in, or yeah, but it is a problem.
Like there are like leaders here in the field, like how come this is not defined yet for us who are just starting in the field to see how, like, what is the right way? Is it number of particles? It is measuring protein. So somebody should like really guide us, and so that we do these experiments in a correct manner, at least.
Yeah, I'm sure many experts are working on that. It's just, I guess, Ken, would you like to have some input on this?
Yeah, so I mean, this is something that the International Society has tried to lead the way on, to at least point out where the differences are, and certainly, you know, we have all these different means of quantifying the amount of EVs, but then we also have to keep in mind that that there are co-isolates in many of our preparations, and so somebody who's doing an ultracentrifugation pellet versus someone who's you know, using multiple methods to purify their EVs are going to come up with a very different mixture of particles. And so I think that it's great to see how Charles and Anthony, how you've used, you know, AF4, for example, to really separate some of these different, at least different sizes of particles. And I see there's a comment here from Scott Ferguson about, well, it's not just the sizes, it's also the molecular composition, you know, so so there I think there are a lot of opportunities here to use reporters like what Charles and Anthony have developed here and tested to try to answer some of these questions and really get at what's happening physiologically. And Esther, did you have anything to add to that? I saw that you were.
No, I think it has pretty much been discussed, and I'm really looking forward to the data on the different subsets. And I was especially wondering this because I also saw that the largest TVs obviously have the largest amount of the signal of the reporter. So I was wondering whether you would still be able to pick up a signal if you would only use the smaller EV types.
Yeah, we are working hard on this, so please give us some time. Thank you.
Thanks.
Thank you.
Well, I think that's a good stopping off point for us, looking towards the future and all of the possibilities that are opened up using this approach that's been pioneered here in different systems, looking at different particles, and hopefully coming up with some biological answers here that will also help us to improve human health. So thank you everybody for joining. A special thanks to Charles and Anthony for their presentation, for sharing their work here. And we look forward to seeing you all again at a future EV Club event. So thanks so much, everybody. Take care.